part 3, Molecules of Life, deals with lipids. And lipids are particularly interesting because they're not one of your run-of-the-mill monomer, dimer, polymer kind of things. Lipids are fundamentally different in how you put them together. In fact, one may wonder why people even put lipids in the same group. They are not true polymers. They are macromolecules, yes, because they're large, but they don't work the same way. And so when people started to get a better understanding of macromolecules and biomolecules in general, they like to pigeonhole things. They like to put things in nice and clean boxes. And so they said, okay, everything that has, carbohydrate kind of things in it, we'll call it carbs. And everything that's protein with amino acids, we'll call that protein. But lipids aren't really so neat in that they don't fit in any particular group. But they all don't mix with water. And so even though just because something doesn't mix with water doesn't really tell you much about its structure or its capabilities, that's how they group them. It's perhaps a strange concept that you group things that are sort of alike but not really. Think about it this way. What if somebody said, okay, I want to group all the things together that fly? And you think, okay, well, wait a minute. Birds, bats, planes, insects? No, you can't do that. Because clearly they don't all belong together, but yet here they are, and they're grouping all the things that don't mix with water together and call them lipids. So that's what we're left with, and that's what we have to deal with. Now, to look at this concept of not mixing with water, we need to get a couple of definitions. Some of these kinds of definitions you're probably familiar with in principle. You know when something deals with hydro, that is probably something dealing with water. When somebody is a bibliophile, then they like books. So this term hydrophilic is somebody who likes water. The opposite of this is hydrophobic, which means they have a phobia of water. So in this case, it would simply be afraid of water. And so these molecules have taken on these terms. Hydrophilic molecules are those that dissolve in water, and hydrophobic molecules are those that don't, such as lipids. Now, lipids include a very diverse group of molecules. Fats are the ones we're familiar with because that is the one that rounds out our food groups, right? We have proteins, we have carbs, we have fats. So that's where that comes in. But it also includes waxes and hormones. Now, waxes are not necessarily things that we immediately think of when we think of humans, but we do have earwax, right? But they are things very important to the surface of plants. So plants, on the surface of their leaves, have waxy layers so that they don't dry out. And that's a lipid, or human hormones. So let's take a look at how we might put these together. The fats we're familiar with consists primarily of triglycerides. And here is another number for you in Greek. Tri. We had mono. That means 1. We had di. That means 2. Now we have tri, and that means 3. So we're getting you a classical education as well as a biology education in this class. Triglycerides. 3 things with a glyceride. What does that mean? Well, we have a combination of something called glycerol. Here's glycerol, and look at what glycerol is. It's basically a hydrocarbon. There is your carbon. There are your hydrogens. But it has these hydroxyl groups, 3 hydroxyl groups. That's glycerol. And glycerol attaches to so-called fatty acids. Now, what are those made of? They are clearly hydrocarbons. This CH2 is C with 2 H's, C with 2 H's, and so on. Clearly a hydrocarbon. And then at the end, it has a carboxyl group. So a fatty acid is a hydrocarbon chain which at one end has a carboxyl group. And so what's a fatty acid in general? How big are these? Well, maybe 20, 30 different CH2s. So they can be pretty long, but they can be shorter, they can be longer, depending on what kind of fat you're looking at. And you take 3 of those fatty acids and you stick them on the glycerol via dehydration synthesis. Remember, here's the water that's coming off. That's the dehydration synthesis part. And this indicates to you that this is a macromolecule because we're building it using the same processes as before. And you end up with a triglyceride, with a fat. What do we need fats for? Obviously, there is a balance in terms of fats and in terms of what we need. Excess body fat is a problem, but fats are needed in the human body. In fact, not too long ago, beginning of the summer, there was an important issue of Time magazine that had butter right on the front. And it turns out that when people started to think about less fat in our diets, that may actually not have been the healthiest choice. And maybe some of you might want to go look back at this, but this craze about low fat and no fat may actually not have been the best. Why? First of all, when there is fat, it tastes better. That's well known, and it's not saying anything about how we taste. It's just that oils tend to carry flavors and aromas better than if they're absent. So that's clearly one thing. But the second thing, and more importantly, is when we do have fat, we take in calories, and if we take out the fat, we replace those calories with something else. In our diets, we tend to replace those fats with carbs. And too many carbs, especially heavily processed carbs, is not good for us. When you think about our digestive system and where we as a human species came from, hunter-gatherers, and what you find outside, you don't tend to find processed carbs. You find sort of a mixture of things. And we have gone away in this time where we're working on the non-fat or low-fat roller coaster, I suppose. This is what everybody, or the pendulum swing into the no-fat and low-fat area. We have lost sight of what we may actually need. And so this article didn't say eat lots of fat. It just said maybe don't cut it all out and watch your carbs. And it is interesting because when that research came out and when people started to work on low-fat and no-fat kinds of products, that coincides with the beginning of our obesity epidemic, which you've probably read about. Now, none of those things are probably due to just this single thing. So also, we get busier, we eat differently, we have fast food. All those things go into that, no doubt. But it is perhaps one piece of the puzzle, and it's just something that people should think about. There appears to be no correlation over time with a reduced fat diet and longevity. People who eat fats live just as long, perhaps even longer, than those who don't. That's just something to note. So what do we use fats for? Well, energy storage is one. Fats do provide long-term storage. Not like glycogen, where you can very quickly get a glucose out. There's no glucose in this, so it takes a little bit more processing. And so fat is really sort of the ultimate storage device, not an immediate storage device. Cushioning. There are many parts of the body that require cushioning, and they require fat in terms of achieving that padding. And if you think of where fat tends to accumulate, around the waist, around the belly, because that's where our sensitive organs are. That is where you would put an area that gives you cushioning. Makes sense. And then lastly, insulation. This is perhaps not so important for us as humans, but there are quite a few organisms that need to have layers of fat so that they have protection against heat loss. So those are some of the functions of fats. Now, the things that make up our fats, the fatty acids primarily, come in different versions. The first of these is unsaturated fatty acids. And an unsaturated fatty acid is one where somewhere in that fatty acid hydrocarbon chain, there's a double bond. So if we look at the carbohydrate chain, at some point there's going to be a double bond, and that's what that is. Remember the double duty of the electrons? And so this is an unsaturated fatty acid. If perhaps you have two double bonds, then it could be a polyunsaturated or diunsaturated fatty acid. So you can have different kinds of fatty acids. And so the way that you put these together dictates to some degree what kind of things these come from. It turns out that just as with the difference between starch and glycogen in the carbs, there's also a fundamental difference about whether you have unsaturated fatty acids or saturated ones. Unsaturated ones tend to be with plants and saturated ones with animals. More about that in a second. Saturated fatty acids are just a straight hydrocarbon chain, no double bonds between carbons. And it turns out that in food processing, one of the things we do is artificially replace these hydrogens in unsaturated fatty acids. What that means is you would take one of these unsaturated fatty acids up here, and you would say, okay, let's not do this. Let's instead just stick the hydrogens on, and we have a single bond here instead of a double bond. We'll give them all the hydrogens they need. But it's artificial. So there's only one bond here, and the hydrogen here, that really isn't supposed to be there. We just stuck it on. We can do that. And the reason why people do this, why we created these so-called hydrogenated fats, is because they help us extend shelf life. Sometimes they stabilize the product. And if you're a consumer, that's actually a good thing. I mean, you like to have fresh things, right? But the problem is, and they didn't know this when they started it, hydrogenation actually makes it into a fat that's bad for us. And if you go back to chapter 1 and look at that research example with the hydrogenated fats, the trans fats, you can follow along and realize how they came up with the research. So here's an example of a triglyceride. And if I showed you something like this, you would immediately see that there is a kink in the molecule, right? There is that kink right here, and that kink is made by a double bond. So whenever you see kinks like this, you realize, aha, there must be a double bond, and a double bond means it's unsaturated. In this case, there's only one, so it's monounsaturated. But there are many different ones you could draw. So let's say here, we're going to draw one, and it has a kink. And it's straight, and it has a kink. Well, this is di-unsaturated, because there's two, right? And then we can have one that doesn't have any. So this one would be a saturated fatty acid, right? So those are the kind of things that you can play with. Now, in animals, as I said before, fats are composed of saturated fatty acids. And you can usually tell because they're solids at room temperature. And if you think about what they are, butter, solid at room temperature, right? Lard, solid at room temperature. And these are the kinds of fats, these saturated fats, they contribute to buildup of lipid plaques, and that's, of course, what promotes heart disease. And so we should have less of those in our diet. And if you look again historically at where the human species came from, having meat was probably a fairly rare event in the life of early humans. And our digestive system is just accustomed to that. We haven't had the fix that makes it equivocal whether we can have fatty acids or not. So right now, having saturated fatty acids in excess is not a good choice. Plant oils tend to have polyunsaturated fatty acids. Those are better for us because they don't create this buildup, and they're liquids at room temperature. Now, they can be wildly different, these plant oils, and it depends on how long these fatty acids are and how many kinks there are. The difference between something like sesame oil and peanut oil, well, they're different seeds, right? And the reason why they're different, these oils, is because they have different kinks and different lengths of fatty acids. That's all it is. In fact, it's very interesting to see, even with olive oil, you have a type of olive that tastes a certain way, and its oil will taste a certain way because it has certain types of fatty acids. In fact, you can even tell a difference between younger olives when they're pressed and mature olives. When those are pressed, you get the virgin olive oil and the normal olive oil, because as the olives age, the composition of these fatty acids changes in the very same olive. So it's very interesting that some of these olives tend to taste better when you harvest them earlier than if you harvest them later. So those are some of the little interesting facts about polyunsaturated fatty acids. Now, if you try to retain some of the taste characteristics of the unsaturated fat, but you wanted the stability of the saturated fat, that is when you would take the kinks out. Take the kinks out, and that is hydrogenation. And this is what creates these trans fats. The hydrogen atoms are added artificially. You can get plant oils that become solids at room temperature, but then it turns out you're actually creating something that's, after all, not good for us. The trans fats are the things that have been implicated in heart disease, and it's simply because we're straightening out these kinks. I mean, this is the kind of thing you have to actually do in order to realize that there could be a problem. You could never say to having this molecule on the table and say, okay, well, I'm taking the kinks out, now it's bad for me. There's no way to actually, so this is nobody's fault. People just don't realize sometimes until much later that whatever it was they invented actually has some negative repercussions. Now, lastly in this group are steroids. And you can see here that steroids are very different in their structure from the fats we just talked about. There aren't any long fatty acid chains. In fact, these steroids are very strange looking by comparison. You have a carbon skeleton that's actually fused to have rings. 1, 2, 3, 4 of them. And they're very tightly bonded together, very solidly bonded together. Unlike the way we had the bonding between the glucose molecules. That was just a single bond. Here it's more. The other thing to see is that cholesterol, shown here on top, that is actually the one that is sort of the ancestral, the base steroid from which all of our hormones are produced. And it's interesting when you look at this, if you look at, for example, our sex hormones, here too, testosterone on the one hand, that's clearly a male sex hormone. And you look at the difference between testosterone and cholesterol, and you realize, okay, we have this difference and that difference, and those are the only differences. Now you go to estrogen, that is a female hormone, and you look at the differences again, and here is your carbonyl, and here's your hydroxyl. So we've taken this and moved it over there and switcheroo. That's all we did. So by just exchanging the two functional groups on these ends here, you end up turning it from a thing that makes you male to a thing that helps you help babies. How very odd, right? It's just a very small change, but it has a very huge effect. And so this is how it is sometimes. With all of these molecules, the small changes are sometimes having great effects. And that's the end of Chapter 3, Part 3.